Hello everyone, my name is Yalbi Balderas, and today I would like to tell you about this research that we have been developing with some friends, and which is entitled Altered Pathways of Hypoxia in Lung Fibroblasts and their relationship with the development of idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis is a progressive, irreversible, and generally fatal disease of unknown etiology. Hypoxia has been described as a determining factor in the development and progression of this disease. However, the role of hypoxia is still unclear. Therefore, the objective of this work was to elucidate the role of hypoxia in the pathogenesis of IPF. Hypoxia and its transcription factors, HIF1 alpha and 2 alpha, influence numerous circuits that could perpetuate fibrosis by increasing myofibroblast differentiation and by promoting extracellular matrix accumulation. Here we aim to elucidate the signature of hypoxia in the transcriptomic circuit of IPF fibroblasts. Well, here you can see our experimental design. A gene expression analysis with 6 lines of lung fibroblasts under normoxia or hypoxia was performed. 3 cell lines were derived from patients with IPF and 3 from healthy donors, giving 36 replicates. We've used the ClarumD platform to analyze the response to hypoxia in both controls and IPF. The macroarray data were analyzed using R packages. Well, then we obtained the results. In figure 2, we can see the response to hypoxia for 48 hours mixing all the control and the IPF fibroblasts. We found that the control response is greater by the number of genes and complexity. We can clearly separate the genes that are responding to each condition. And the gene biological processes are the classic response to the lack of oxygen. We use Ingenuity pathway analysis software to know specifically how they are responding to hypoxia. Then we decided to analyze each cell line separately, and we can see that there is a high heterogeneity between cell lines. It is clear that there are some common genes responding to the low oxygen. We finally got a signature of 35 genes that respond to hypoxia in both controls and IPFs, but there are some genes that are specific for those groups. For example, there are 19 genes that are exclusive to IPF. In this table, there are the genes that participate in IPF, so you can check it later if you want to know more about this project. Among the genes involved, EPAS1, that is HIF2 alpha, plays an essential role in this chronic adaptation to hypoxia. Finally, we conclude that the analysis of the enrichment of the 35 differentially expressed genes shared in both groups reflects a general response to hypoxia. The enrichment of the 311 differentially expressed genes in the controls gives us information on the response of the healthy control fibroblasts to hypoxia and response that the IPF fibroblasts do not have. Furthermore, what it is observed with the analysis of the enrichment of the 19 differentially expressed genes in IPF are the exclusive pathways that could be potential therapeutic targets. Among the genes involved, HIF2 alpha plays an essential role in this chronic adaptation to hypoxia. We want to acknowledge the funding received from CONACYT and thank you for being here. If you have any questions, you can ask me on this platform. Please send me any comments you have, or maybe you just want to send me a like. Thank you. Bye.